future medical professionals. Welcome back to another episode of Biochem Breakdown, proudly brought to you by Antibodies Review Center, your go-to space for simplified, solid, and smart review sessions. I am Teresa Austria, your guide for today. So today's topic is one of the most exciting players in protein synthesis, the transfer RNA or tRNA, and its role in translation. So before we begin, please ensure you have your pen and paper. And now let me ask you, have you ever wondered how your cells know which amino acid to place and where when building proteins? So today's topic is the transfer RNA or tRNA and its role in translation. So here's what we'll cover: an overview of protein synthesis, the structure of tRNA, anticodon and codon pairing, tRNA interaction with ribosomes, amino acylation or charging the tRNA, and finally the role of tRNA in the translation process. Here are our objectives. By the end of the lesson, you will be able to define transfer RNA and identify its key structural features, explain the role of tRNA in translating mRNA codons into amino acids, demonstrate correct codon-anticodon pairing and its importance in protein synthesis, and illustrate how tRNA interacts with the ribosome during the three stages of translation. Before we begin our lesson on tRNA and its role in translation, let's warm up with a short pre-test. Don't worry, this isn't graded. So take your time, read each question carefully, and just do your best. You'll have 15 seconds, or you can pause the video if you need more time. Ready? Let's begin. So the first question is, what is the main function of tRNA during translation? A. To carry DNA instructions, B. To synthesize mRNA, C. To bring amino acids to the ribosome, or D. To form peptide bonds. The second question would be, the three-letter sequence on tRNA that pairs with a codon is called A. Codon, B. Anticodon, C. Ribosome, or D. Transcript. So the correct answers would be, what is the main function of tRNA during translation? The answer is C. To bring amino acids to the ribosome. And for the second question, B. Anticodon. Alright, great job answering the pre-test, everyone. Don't worry, if you found some of the questions a bit challenging, that's okay. Now, before we dive deeper into the role of transfer RNA or tRNA in translation, let's first take a quick overview of how protein synthesis works as a whole. This will help us connect today's topic with a bigger picture. Protein synthesis is the process by which our cells build proteins, which are essential for life. This process follows the central dogma of molecular biology, which says DNA to RNA to protein. These are two main steps in protein synthesis. First, in transcription, which takes place in the nucleus. DNA is used as a template to create messenger RNA or mRNA. RNA polymerase is the enzyme that helps in this process. mRNA carries the genetic instruction from the DNA to the cytoplasm. The second step is translation, which happens in the cytoplasm at the ribosome. The ribosomes read the mRNA sequence in codons, the three base codes. Transfer RNA brings the correct amino acid to match each codon using its anticodon. As amino acids are added one by one, a polypeptide chain called protein is formed. So tRNA plays a central role in translation. It ensures that the genetic code is accurately translated into a protein. Now about the structure of tRNA. It is a small single-stranded molecule of RNA that transfers specific amino acids to the growing polypeptide chain at the ribosome during protein synthesis, or what we call translation. The main role of tRNA is to deliver the correct amino acids to the ribosome by matching its anticodon with the codon on the mRNA, ensuring that the protein being built has the correct amino acid sequence. The secondary folded structure of tRNA has three hairpin loops, giving it an appearance of a three-leaf clover. The main constituents of tRNA includes the acceptor stem at the 3' end, which ends with a CCA sequence and is the site where the amino acid attaches. The anticodon arm, which contains the loop with a 3-base anticodon that binds to the complementary codon on mRNA. The D arm containing the hydroridine bases that helps maintain tRNA structure and recognition by amino acid tRNA synthase. The TC arm containing ribosymidine, pseudoridine, and cytidine, important for ribosome variable loop, which varies in length and sequence that adds flexibility to tRNA. Regarding an anticodon pairing, a codon is a sequence of 3 nitrogenous bases on the messenger RNA. Each codon corresponds to a specific amino acid or a stop signal in protein synthesis. An anticodon is also a 3-nucleotide sequence, but is located on the transfer RNA. The anticodon is complementary to a codon on the mRNA strand. For example, if the mRNA codon is a 5' AUG to 3', which codes for the amino acid methionine, the corresponding tRNA anticodon would be 3' to UAC to 5'. You might remember that in mRNA, adenine pairs with uracil, thymine with adenine, cytosine with guanine, and guanine with cytosine. But in translation, the pairing between mRNA and tRNA, the bases pair using standard complementary base pairing rules. Adenine A pairs with uracil U, and cytosine C pairs with guanine G. Here's an example to go through step by step. Suppose the mRNA codon 5' CGA 3', the tRNA codon matching would be 3' GCU 5'. Using a codon chart, CGA codes for arginine, ARG or R. Before we dive into the next topic, let's take a quick checkpoint to test what you've learned so far. Feel free to pause the video if you need more time. Let's see what you've got. So we have 3 codons, and we need to find its anticodon and its corresponding amino acid. So firstly, it's AUG, and then for letter B we have GGC, and then for letter C we have UAC. Time's up. Let's check how you did. Here are the correct answers. For letter A, the codon AUG would have an anticodon of UAC, which has the amino acid of methionine. For letter B, we have GGC, which corresponds to an anticodon of CCG that has an amino acid of glycine. And lastly, for letter C, you have UAC, which has an anticodon of AUG, which corresponds to the amino acid tyrosine. If you got them all right, great job. If not, don't worry. We're here to learn and improve. Now that we've cleared that up, let's move on to the next part of our discussion. As we can see, we have a ribosome composed of 2 subunits, large and small. The ribosome has 3 active sites, the E site, the P site, and lastly the A site. To remember easily, A site is for the arrival of new tRNA, P site is for the peptide bond formation, and E site is for the exit of uncharged ribosome by matching its anticodon with the starting codon, AUG, on the mRNA. Next, incoming tRNAs carrying amino acids enter the A site, where their anticodon pairs with the corresponding mRNA codon. A peptide bond forms between the amino acid at the P site and the one at the A site. The ribosome then shifts, moving the tRNA from the A site to the P site, and the anti-tRNA moves to the E site to exit. This cycle repeats elongation the polypeptide chain until a stop codon is reached. Before translation begins, each tRNA is charged with its specific amino acid by an enzyme called amino acyl tRNA synthetase. This process uses ATP and ensures each tRNA carries the correct amino acid based on its anticodon. For example, a tRNA with the anticodon UUC will be charged with lysine, because UUC pairs with the mRNA codon AAG, which codes for lysine. Now let us proceed with the process. So first would be translation. This occurs in 3 steps: initiation, elongation, and termination. So during initiation, translation begins at the AUG start codon on the mRNA. The small ribosomal subunit binds to the 5' end of the mRNA, scans for AUG and the initiator tRNA or tRNA met carrying methionine, which binds to the P site of the ribosome using its UAC anticodon to pair with AUG. The large ribosomal subunit then joins to form the complete ATS ribosome in the eukaryotes. Next would be elongation. This is where most tRNAs are involved, and the cycle repeats for each new amino acid. First, a new tRNA charged with its amino acid enters the A site of the ribosome. The tRNA's anticodon must complement the codon on the mRNA. For example, if the mRNA codon is GCU, codes for alanine, the tRNA codon is CGA. Then, the ribosome catalyzes the formation of a peptide bond between the growing polypeptide held at the P site and the new amino acid at the A site. The peptidyl transferase activity that catalyzes this is a function of rRNA, a ribozyme. Next, the ribosome shifts by 1 codon. The tRNA in the P site moves to the E site, or exit. The tRNA in the A site moves to the P site, now carrying the polypeptide chain. A new tRNA enters the A site, and the anti-tRNA in the E site is released. This cycle continues with tRNA molecules entering, delivering their amino acids, and during termination, when the ribosome reaches a stop codon, UAA, UAG, or UGA, no tRNA binds to it. Instead, it releases factors, binding to the stop codon and trigger the release of the newly made polypeptide chain, disassembly of the ribosome, and recycling of tRNA and mRNA. Looks like we've got a checkpoint. Let's see how well you're keeping up. You have 15 seconds, or feel free to pause the video if you need a bit more time. Give it your best shot. Number 1. Which step of translation involves the ribosome assembling around the mRNA and the initiator tRNA? Is it A. Elongation, B. Termination, C. Initiation, or D. Transcription? Number 2. During elongation, what happens at the A site of the ribosome? Is it A. The polypeptide is released, B. New tRNA with an amino acid enters, C. mRNA exits the ribosome, or D. Ribosome disassembles? Number 3. What triggers the termination phase of translation? Is it A. Mutation in the gene, B. A ribosome running out of GTP, C. A stop codon appearing on the mRNA, or D. An uncharged tRNA entering the A site? And that's it. Let's see how you did. So the correct answers are, for number 1, which step of translation involves ribosome assembling around the mRNA and the initiator tRNA? The answer is C. Initiation. For number 2, during elongation, what happens at the A site of the ribosome? The answer would be B. New tRNA with an amino acid entry. And lastly, number 3, what triggers the termination phase of translation? The answer would be letter C. A stop codon appearing on the mRNA. Whether you nailed it or not, you still had a few confusions. That's okay. Progress is what matters most. You've made it through this checkpoint, and that's already our references to sources that help shape the ideas shared in this video. Before we end today's lesson, I would like you to answer a short formative quiz titled Decode the Message to assess your understanding of transfer RNA and its role in translation. So for the instructions, to begin, simply scan the QR code shown on your screen using your device. This will take you directly to the Google form. In this activity, you will be given an mRNA sequence to decode. From this, you are required to write the correct tRNA anticodons for each codon in the sequence, identify and list the correct amino acids in order, indicate which tRNA enters the ribosomal sites, may it be A, P, or E, at each step. And lastly, mark the start and stop codons to show where the translation begins and ends. This quiz will consist of 3 items, and after submitting, your score will automatically appear on screen for immediate feedback. Make sure to review the lesson content as needed before answering. Alright, that's a wrap. Hope you got good dose of knowledge without getting too much at once. Don't forget to check in with your Antibodies Review Center whenever you want to brush up. This has been Teresa Osia, and catch you later.